Hello, everyone. My name is Egina Francis Makwabe. I'm the consultant nephrologist in Tanzania, working with Africa Healthcare Network. Together with Dr. Ntalindwa Joseph from Rwanda. He's a consultant nephrologist and physician working with AHN Africa Healthcare Network in Rwanda. So we'll be moderating today's session. The AHN Nephrology Educational Webinar, series number 51. The topic today is understanding the HLA system in organ transplantation. So it's my great pleasure to introduce to you today's speaker, who is Dr. Shruti Tapiawala, who is a senior consultant nephrologist and a renal transplant physician, Institute of Renal Science and Global Hospital, Mumbai, and Sir H. Reliance Foundation Hospital and Renal Research Center in Mumbai. She's also a director, Advanced Transplant Diagnostics Laboratories, PVT Limited. Dr. Shruti has a fellowship in clinical nephrology and transplantation, University of Toronto, Canada. And also she has a fellowship in transplant immunology from the University of Toronto in Canada. So Dr. Shruti, you are warmly welcome. It's a pleasure to have you here today. And we are, ready to listen from you.
Okay, should I start sharing the screen?
Yeah. Yes, please.
Okay, so good evening, everyone. And thank you. First of all, I would like to thank Dr. Vincent because, it has been a pleasure. I know him since, I think, more than 15 years now. And I know that he's doing a great job out there. And I knew him right from there where, he would be working in the transplant department and in the hospitals. And, it was great working with him. And this is a welcome opportunity for me to share my experience and to discuss about HLA system and organ transplantation, at least what I have learned and what I'm practicing in the country. And I'm in touch with most of the people back in Canada because I want to see that the advances keep happening and I can see that it goes along in our country and to spread it more. So let me start. We all know that organ transplantation is a medical procedure in which an organ is removed from one body, placed in another body of a recipient to replace a damaged or a missing organ. It is one of the greatest advantages of modern science and one of the best options as we know for organ failures. At least for kidneys, we know that dialysis is not a good option and it can only be a life-saving option. But the best option still remains transplantation from economical point of view, from medical point of view, and from the longevity point of view. A transplant organ we will call as a graft, and transplantation goes along with the graft. What are the types of grafts that we know? Now, I have made this into a very basic lecture because I would understand that there are students also attending. So my apologies for senior nephrologists who will find this a little basic, but going ahead, I will try to see that I cover most of the type of audiences listening to this. So when I look at the type of grafts, I would think that there are few types of grafts which I need to understand when I'm addressing transplants. One is an isograft, which means that it is a graft between identical twins. This is the type of transplant which happened in 1957 when Dr. Joseph Murray transplanted the twins, and that was a first successful transplant in the history of science. Then comes autograft, where you are grafting in individual's own organ into the same body. So that is an autograft, say skin transplanted from one area to the other. Then is allograft. That means you are transplanting organs between the same species, but not identical twins. So human being to human being, but of a different genetic profile. It is non-identical. It becomes an allograft. Between two different species is a xenograft, wherein you have an animal organ given to a human organ. So we have a famous deity known as Ganpati, where we give an example saying that this was a xenograft, but that was an idea which came about from ancient Indian medicine, where it was thought that if one part of your body is spoiled and it is not working and damaging you, you better replace it. So that was an idea which was put forth, and I would say that modern science has not been able to do that as yet. So what are the immune responses? Why we cannot do it? So transplant rejection is caused by the genetic differences between the donor and the recipient. Hence, the isograft worked wonderfully without having any modern medicines. It worked only with the steroids, and at that time, maybe it was cyclophosphamide and it was different drugs which were very toxic. But because it was an isograft, it was between identical twins, the response and the genetic differences were not much. So we look at HLA antigens and we look at blood group antigens, because these are the differences which our body is going to see and react to. Alloantigens, this is a terminology which is used for antigens which vary between members of the same species. So if you're not identical twin, then your antigens are going to vary. Alloreaction, the immune response to an alloantigen is known as an alloreaction. Alloreactions can be two kinds. One can be host versus graft, which is our typical transplant rejection, that our body is mounting a response to the graft. And the second is graft versus host, which we usually see in the bone marrow transplantations, where the bone marrow is now acting up against the body's immune system. So there is a difference between the types of reactions which we see. So when a solid organ is transplanted, you see a host versus graft. And when you see a bone marrow transplant rejection, you might see a graft versus host disease. Now, what are the effectors of rejection which we see? So the major players in this game is T cells, B cells, antigen presenting cells, and MHC. MHC, major histocompatibility complex. This is the most important, because this is where the differences between two individuals is identified in the recipient. So the T cells, as we all know, arise in the thymus from the bone marrow derived precursors. Each T cell has a unique T cell receptor, which is a clone. There is a positive selection, there is a negative selection, and there are CD4 and CD8 subtypes. CD4 subtypes will be antigen-specific immune response, and CD8 will be precursors to the cells and class 1 MHC. B cells will arise from a mature bone marrow, and it will mature in the bone marrow. There will be negative selection. There will be expressions of B cell receptors on their surface. When these are simulated, the B cells will secrete antibodies of the same specificity as their receptors. What are antigen presenting cells? Most important, because these are the cells which are going all around your body and presenting the antigen. And these are the antigen antibody complexes which are going to then go ahead and cause a reaction. These activate the T cells, they endocytose the antigen, display it on the MHC molecules, then the T cells go and recognize, interact with the antigen MHC to become activated. So now we go to the HLA system, which is the basic or the backbone of transplantation, because if we understand the HLA system well, we know how it is working and how we can proceed with the transplantation. So the genetic loci involved in the rejection of the foreign organs are known as major histocompatibility complex. And highly polymorphic cell surface molecules are encoded by this MHC. It is called the HLA system because it is a human leukocyte antigens, because these antigens were first identified and characterized using allo antibodies against the leukocytes. Hence, it is known as human leukocyte antigen. Agglutinating antibodies were observed in the sera from multiparous women and previously transfused patients. So this was how they found out that, okay, when a human being is exposed to different genes or different proteins, then these are made into antibodies. The reaction makes antibodies in our system. And that is how they understood that if these are antibodies which our body has made, then the antigens coming must be different. And then the HLA came into existence. Graft rejections were found to be associated with development of antibodies against these allogenic leukocytes. So initially it was known to be the cells which are causing it, and then as we went ahead and we observed and we identified, we understood that it is the cell surface and the encoding of the genes which is giving you the differences. The HLA system has well been known for transplantation antigens, but the primary biological roles of the HLA molecules is in regulation of the immune response. So the HLA exists on all nucleated cells in the body, mainly class 1, loci A, B, and C, on all the cells including T cells. Class 2, loci DR, DP, and DQ, and antigen presenting cells, B cells, dendritic cells, but not T cells. The purpose is to present an antigen, foreign class 2 and antigen endogenous, in its groove to the T cells in order to generate an immune response. So we have to understand how it does. It also identifies self from non-self. So this self from non-self is the most important thing which our body should not see. If you have a self antigen coming in, your body is going to accept it as its own. If you have a non-self coming in, if given a chance, it will react, it will make antibodies to it, and those antibodies are going to be a force which are going to attack the graft if that non-self is seen again. So which is a very important thing to keep in mind. Location and organization of the HLA complex is on the chromosome 6. So the short arm of chromosome 6 is where all the work is. So you have class 1, class 2 and class 3. Class 3 are not very important for organ transplantation. So we are going to look at class 1 and class 2. Now if you see, there are MIC-A and MIC-B antibodies also. These are minor chain related antibodies which are important in, say, heart transplants, lung transplants, but less in kidney transplants. But sometimes there are reports which say that even these MIC-A antigens are important. So now if I localize it, that the short arm has class 1 which has A, B and C loci, class 2 as DP, DR and DQ loci. So for me as a transplant physician, as of today's modern science, I know that A, B, C, DP, DR, DQ have to be seen. I have to see what is self, I have to see what is non-self when I give a particular donor to a particular recipient. Whatever is non-self, I have to try to see that will this body react to this patient. Very important. So 3 types of MHCs. Class 1 encodes glycoproteins expressed on the surface of nearly all nucleated cells. The major function of the class 1 gene will be to present peptide antigens to the cytotoxic T cells. Class 2 will encode glycoproteins primarily on APCs, that is antigen presenting cells. Example, macrophages, dendritic cells, B cells. They will present antigenic peptides to T helper cells. So they will more act in the indirect pathway. Class 3 will encode for various secretory proteins and that have immune function, and they will be all the destructive system like the complement system, factor V, TNF, which are molecules and which are the effectors of the inflammation. So this is how you can visualize it, that the RBCs usually do not have most of these class of proteins. The nucleated cells and the APCs are in action. So the function of both class 1 and class 2 is to present offshore pathogen-derived peptides to the T cells, a process that will initiate the adaptive responses. Class 1 sample cytosolic proteins and detect the foreign proteins that would then indicate an intracellular pathogen such as virus or intracellular bacteria. In this case, it's a gene which has come from with the allograft. Recognized by the CD8 cells, which provide a surveillance system to target the infected cells or the cells with that particular antigen for destruction. So this is the type of expression which I showed you, that these are the chains. Class 1 has alpha 1, alpha 2, alpha 3 and beta 2. Class 2 will have alpha 1 and alpha 2, beta 1 and beta 2. Okay, so these are the grooves. If you go to see, this is a popular image from NEJM where you have the peptide grooves which are being seen, where the antigen presenting cells can engage, or the antigen can engage to the antibody can engage to this groove. Now that means that class 1, class 2 are the most important and relevant to our transplantation, because this is the site of action. So I would want to check class 1 typing A, B and C, because that is the HLA typing is going to give me the identification of the loci, antigens which are present on the donor and on the recipient. Class 2, DR, DP and DQ, because these are the most important loci or the antigens which will present a challenge or a stimulus to form antibodies in the recipient if they are mismatched or they are foreign. So mismatches are a matter of immunological risk. Mismatches also impact the short-term and the long-term survival, because more the immunological risk, more the mismatch, the more stimulus you're giving to that patient's body to make antibodies. This is a famous study where in 1995, Dr. Terasaki showed that, the better the match, the survival is the best. So if you see, maximum mismatches will have the lowest survival. So 6 mismatches, that time they were doing only ABDR because they did not know that other antigens were also. 0 mismatches did the best. That is the highest survival. And the lowest survival is that of 6 mismatches. So we can imagine, and that is how people understood that more the antigen matches, it's better. But obviously in today's world and in the practical situation, we do not find twins. We do not find complete matches. But that doesn't mean we keep patients on dialysis and we say that, oh, your outcome is going to be inferior, we will keep you on dialysis. But dialysis outcome is far more inferior to transplantation, is well proven. So HLA typing information is required for matching. It is required for future complications. So if you have rejections in the future and you want to know that why the rejection happened, or is there an antibody against the donor, if we do not have the donor typing, it is very difficult to understand that whether the antibody in the patient system is against the donor or not. Information required to understand if a prospective transplant recipient has antibodies. So even pre-transplant, it is important for me. Post-transplant, it is important for me. And the degree of match tells me how much immunosuppression I am supposed to use and what I am supposed to expect in the future. Hence, HLA typing forms the most important aspect of a transplant. Most of the times I had seen few years back that, the nephrologist would say that, oh, HLA typing is a little costly. I'm only going to do a cross match because all that matters is a cross match. But the typing was only seen as to be done for the matching point of view and not from scientific point of view, because they wanted to just prove the relationship and go ahead. But that is not what. Scientifically, you need it, because that is going to help you manage the patient throughout the transplant career of that patient. It can be serological, which is an old method. I don't know what Africa uses, but serological method has now progressed to molecular method, which is more precise, because we have more and more HLA antigens which have been identified, and on those antigens, more and more amino acids, alleles have been identified. So the recommendations are, as of now, to do A, B, C, DR, DP, DQ, because these are all implicated in the short and the long-term survival. Now, how do you understand what is written in the HLA typing? So historically, if you look at the serological typing, how did the nomenclature come by? So it was not that the HLA antigens came labeled. We started labeling the antigens based on their order of discovery. So if A1 was found first, it is A1. After that, whatever was found was A2 and then A3 and so on. Rule of the thumb, the lower the number, the common the antigen. Very important. Because if the antigen is very common, then you will see it in a lot of your patients. And if they have antibodies against a common antigen, the transplantation is going to be very difficult. Because then that patient will have antibodies against a lot of population. Molecular techniques. So you have molecular techniques where you have HLA, where you have a labeling, where you see A with the asterisk. So when you see A with the, you see an antigen number or an antigen alphabet with an asterisk, you know it is a molecular type. Post the asterisk, you will find antigen number, which will always have a 0 in front of it. Then you will have the allele. And then you will have more ramifications. If it is a next generation sequencing and a bone marrow transplant, you will have a longer string, which will tell you the amino acid residues on it. So numbers from the serological and the molecular change, because as our complexity of knowledge changes, the numbering has changed. So now it is suggested that most of the labs all over the world should move to molecular typing, because that is how the antibody profiling is given to you. So you can match apples to apples, because most of the other technology is molecular based. So now this is an example more for students, that if you have a donor which has this profile, and you have, if you have a recipient with this profile, you can see that the recipient has A2, A32. So the donor A24 is going to be seen as foreign. The recipient has B35, B51, identical. Recipient has DR1, DR13. Both the donor antigens are going to be mismatched. Now, if you reverse it, if you reverse the donor and the recipient typing, what happens? If you have the donor as A2 and A32 and the recipient as A24, you have two potential stimuli going from A locus. B remains the same, identical. DR remains the same, 2 locus. So see how it changes. So you have to check what is there on the donor, which is different from that from the recipient, and that is what is going to be the antigenic stimulus. Same way, A24 over here is homozygous. That is why you see a dash over here. So that means if this recipient has any antibody against A24 and this donor is given to this recipient, what will happen is that there will be a double stimulus from this, and there will be a bigger magnitude of rejection. So what are rejections? Rejections or reactions of our body to the graft. So it can be acute cellular rejection or it can be an antibody-mediated rejection. So differences in the HLA antigens, mismatches will incite the reactions as the body recognizes non-self on the mismatched antigens from the donor. Cellular rejection, usually caused by T-cell mediated reactions. Destruction of the graft occurs by the CT8 cells and the CD4 helper cells. Pathways can be direct and indirect. Now this is a very basic knowledge and more for the students, that the direct pathway is where the antigen presenting cell in the graft is directly engaging with the antigen, the antibody which is there rotating in the system. And it is going to activate the CD4 and the CD8 cells, and it is going to cause endothelitis, tubulitis, and damage. What is the indirect pathway? That the recipient's antigen presenting cells is processing the antigens, presenting it to the surface, and then that is engaging with the CD4 cells and causing the reaction. What will also be targeted is that the antibody secretion will start and they will form clones. There might be a plasma cells which will cause a continuous supply of antibodies, and there can be a chronic or a mixed type of an allograft rejection. So antibody-mediated rejections, also known as humoral in the past, vascular or antibody-mediated, they can be hyperacute. That means on the table. I don't know how many people in this audience has seen it, but as student, I have seen few hyperacute rejections where when we transplant the kidney and anastomose the kidney and the clamps are released, the kidney turns blue on table. So even before you close up the recipient, you can see that the kidney is blue, cyanosed and gone. That means the rejection happened immediately and there was a force which was ready to attack the kidney. Presence of preformed donor-specific antibodies can do this. So if your body or the patient's body already has antibodies against the allograft which is coming in, the moment you allow the blood to go into the allograft, the antibodies are going to engage with the antigen on the allograft and cause the rejection there and there. Acute, where you have initial exposure of the HLA-1 and 2 antigens in cases of, say, previous transplants or previous transfusions, pregnancies, etc. There, the antibodies will cause a complement-dependent cytotoxicity, inflammation, and cause a rejection in an acute manner. And this we usually see when we have patients who are very sensitized, and by 8-day, 9-day, or 1 month, we see these rejections. So what are anti-HLA antibodies? So it is long recognized that these antibodies reacting with the HLA antigens which are expressed on the renal allografts, specifically to the donor, they will cause hyperacute rejection, acute vascular, and chronic rejections, which will result in graft failures. Anti-HLA antibodies will develop following exposure to HLA antigens through blood transfusions, pregnancies, and previous transplants. Why? Because these patients have been exposed to different HLA antigens from the same species but different genetic. Now, what else do you have to look at antibodies? One, you saw that whether they are already formed into the system. They are formed because they are already exposed to those antigens. Now you also have to see from the timeline point of view. So if you have a transplant happening today, you have to see in the current serum whether there are antibodies which might cause hyperacute rejection. Historically, did you have those antibodies? Now this is a very important concept, because suppose you got a blood transfusion 15 years ago, and that blood transfusion was from a person who had antigens similar to the donor who is giving you an allograft. That means one of the antigens, say A24 was already seen by your body, which is not self. It formed antibodies and kept, that is historic antibodies, and they are there in the memory. Now you got an allograft and that memory is evoked. So the historic antibodies are preformed antibodies which will come out and cause an acute rejection. Current is where your body has circulating antibodies against the allograft which is coming, and it will attack immediately and cause an accelerated or a hyperacute rejection. Post-transplant, you may develop new antibodies or a memory response. So the memory response is this historic antibodies which came out and damaged the allograft. De novo means newly formed antibodies. That means you got an allograft from someone else, and that allograft is now inciting a stimulus and your body is forming new antibodies which were never there in your system. So that is de novo. So over time, you can develop de novo antibodies. Hence the importance to identify both preformed antibodies and monitor for de novo antibodies or a memory response. Because remember, today I may not have seen the patient 15 years ago. How do I know that this patient has a memory for these antigens? So it is always better. We internationally are now doing it. Nationally also we are doing it, that anytime a patient presents to you and we are working up the patient for transplantation, what do? We test them for antibodies and keep testing them few monthly, so that anytime there is an antibody surge in the body, we can record them and keep, so that we can use it in the future to our advantage in helping the patient having a low immunological risk. So like I said, antibodies are important because they sometimes increase the wait time to receive an organ, and there may be death on the waiting list because sometimes patients have had so many antibodies, antigen exposure, that they are highly sensitized, and they always will cause more rejections, and they will always have cross matches positive and prevent them from getting organ. These antibodies will cause hyperacute and acute rejections. They will cause chronic rejections. How will they cause chronic rejections? It will happen that low level of antibodies might keep smoldering in the system, and they might keep damaging the organs which have been grafted, be it heart, be it lungs, be it kidney. So it can be transplant glomerulopathy, it can be Boop-like syndrome, it can be coronary artery disease which can come in a heart allograft. So we have to be very wary about these antibodies, because even in low titer, they can keep burning the tissue and cause inflammation, which can cause injury. Now, this is a neat study which was done where they found the impact of preformed donor-specific antibodies. So if you see the first graph, you can see that if you do not have any antibodies, so anti-HLA antibodies are negative, but these donor-specific antibodies are also negative. Now, which is donor-specific? So you might have a lot of antibodies, but few of them may be specific to that donor. So you have to understand the difference between the HLA antibodies and what is donor-specific. So if you have no HLA antibodies at all, that means you do the best, because there is nothing to attack the allograft. Second, you have anti-HLA antibodies, but there are no donor-specific antibodies, then you do the second best, but you still have some harm happening, because that means your body is more prone to form antibodies and they might land up killing the allograft. Third is where you have anti-HLA antibodies and they are specific to the donor. So your body already has antibodies which are specific to the antigens coming with the donor allograft. So then that is the most detriment. Second is we are trying to check if class 1 is bad or class 2 is bad. Now here also again, the first graph is where you do not have any donor specific antibodies, you do the best. The second and the third lines, the graphs which are shown, are class 1 and class 2. So it is shown that statistically nothing is very significant whether you are class 1 antibody or class 2 donor-specific antibody. Both the ways, your graft is having an inferior outcome. With the donor-specific antibodies, if you get an AMR, your graft fails rapidly. But with the donor-specific antibodies, if you do not get an AMR and you have managed the immunosuppression nicely, your graft will still last. And overall, if you have DSA negative versus DSA positive, no brainer, the DSA positive will have an inferior survival. So pre-existing HLA antibodies will form with previous pregnancies, with blood transfusions, previous transplants, cross-reactivity to viral antigens. Say influenza virus is a big inciter for inciting a cross-reactivity and forming antibodies. So which we have seen, I have seen in studies when I was in the lab, that in flu season, people would have a surge of antibodies. So we would actually collect PRA samples at that time, so that we catch hold of the cumulative list of antibodies the patient has. And this is called sensitization, and it is very important to take a sensitization history in any and every patient who is going ahead for transplantation. Now that means that I will need to identify the antibody. Very important. What are the methods we have? A cross match. So the first three panels are showing you cross match. A CDC cross match, that means a complement dependent cytotoxicity cross match. Anti-human globulin enhanced CDC cross match, a flow cytometry cross match. So these are cross matches. I will in some time just describe these to you. And the last panel is a flow solid phase, where if you see the difference, suddenly there's a transition from a cell to a bead. So here you are seeing that there's a cell which is being used for the test. And once the flow cross-match panels are over, you can see that there are beads, which are factory-made beads, which are being used to check for antibodies. Now let us see what is the difference. Let us understand what is a cross-match first. These are the broad methods with which we identify the presence of HLA antibody in the patient. So cross-matching was developed in attempt to identify recipients who are likely to develop acute vascular rejection of a graft from a given donor. So it is donor specific. This phenomenon of hyperacute rejection is the result of preformed antibodies to one or more HLA antigens of the donor, referred to as donor specific. So if my body has antibodies specific to the donor allograft coming in, my cross-match should turn positive, because I want to identify these antibodies, and that is why the cross-matching test was developed. It was developed way back in 1969, where it was seen that if you have a positive cross-match, then the immediate failure chances are 80%. Without a cross-match, 26% immediate failures were seen. With a negative cross-match, 15% immediate failures were seen. Recipient without any anti-HLA antibodies, only 2.4% immediate failures were seen, but they were seen. So it was finally concluded in this landmark paper that a CDC cross-match or an HLA cross-match, it was called at that time. If you have a positive cross-match, it was a definite big capital no, do not go for transplant, because if you go ahead with a positive cross-match, 80% chance that you will lose the allograft. After this paper, until 10 years ago, the CDC cross-match remained a robust test to take a transplant ahead. It still remains a robust test, but now we have more technology to add to it. What does the CDC do? So simple, it is a mini transplant which is done in the lab. All that we do is we take the donor cells, we will incubate it with the patient's serum. Why serum? Because serum has antibodies in it. If the serum of the patient has antibodies against the patient's HLA antigens, they will be present on the cells of the donor. They will attach. Once there is an engagement, as you see in this diagram, the blue one is the antibody and the green one is the receptor or the T cell receptors which are there. Once this engagement happens in our body, there's a complement system which is coming into action. So we add a complement to add fuel to the fire. Once the complement is added, the cascade goes through and the cell or the allograft, so-called experimental allograft, is destroyed. And the cells get holes on the surface, is a very common word we use. The cell membrane is lysed. When the cell membrane is lysed, it is open to let everything inside. So then we put a vital dye. You put a eosine dye in it. So when we look at a cross-match under a microscope, what are we looking for? We are looking for dead cells which have taken up the dye. So if the dead cells is more than 20%, we call the cross-match positive. We then say that the patient has HLA antibodies against the prospective donor. Do not proceed or proceed with caution. So that is a CDC cross-match for you, complement-dependent cytotoxicity, because we are adding a complement and we are helping the cell to get lysed if there is an engagement of an antigen and an antibody. Let us see a case. A 43-year-old male, remote blood transfusions, 4 antigen mismatch, deceased donor, 1 DR and 1 A match, current and historical PRA. Why historical? You want to look at the previous PRA also. By CDC. Now see, this was not a solid phase, it was CDC, because in the previous world, only cell-based assays were available. We did not have advanced technologies. So this is a case from before 2000 and before maybe 2000s. The CDC cross-match was negative. It was decided that this is a low immunological risk. Decision to go ahead with transplant done, but within 24 hours, no function in the allograft. Many of us have gone through this situation. I'm sure the senior nephrologist in this group will recognize this. Patient taken back to the OR, histopath is showing severe rejection, antibody mediated. See, arteritis, tubulitis, glomerulitis, lysis, and the kidney had to be removed. This is a C4D which is strongly positive. So what happened? We saw that the CDC cross-match was negative. Why did the patient get rejection? If we have to look at this study, negative cross-match, 15% will get a rejection. So that means this is not very foolproof. There is some problem. So what are the drawbacks? Not all lymphocytotoxic antibodies that cause a positive cross-match are specific to the donor HLA antigens. Some antibodies will display autoreactivity, causing lysis of patient's own cells. So then you get misled. There might be a false positive cross-match and we might prevent someone from rejection. At the same time, you might have a negative cross-match and hyperacute rejection may happen, like it happened in this case. There may be cases where the antibody levels are so low that the CDC will not detect it. So these are major flaws of a CDC cross-match, but still we cling on to the CDC cross-match because that still remains the backbone. So this was a study again by Dr. Terasaki, who developed the cross-match, CDC cross-match, where in 2007 he has published this, that very low titer of antibodies are associated with acute rejection, and these will not be picked up on a CDC cross-match. If you ask me the amount of antibodies required for a CDC to turn positive, it is more than MFI of 20-30,000. So that means you need really high levels of antibodies for the CDC to turn positive. So that means we might be giving so many CDC negatives where there are antibodies floating around in the system. So cross-matching in transplantation went through a lot of technological advances because the CDC was not enough. CDC was then made enhanced by adding anti-human globulin, but still it was not enough because still low titers were being missed, even though it was better than the basic NIH CDC. Then we started doing auto CDC, allo CDC. We started cross-matching with patients' own cells also to look and differentiate auto antibodies and allo antibodies. So this is now done routinely. Next came the flow cross-match. The flow cross-match was a very sensitive platform, and there we were not able to identify this is really positive or negative, which I will show you what is a flow. And last is a virtual cross-match, which is a technology-bound cross-match. Virtual means we are doing it in thin air, but it can be very robust. The technology is robust. So what is a flow cross match? Again, the same principle, a mini transplant experiment in your lab. You have a donor cell. It is incubated with the recipient serum. Instead of adding a complement, you are adding an. So these are secondary antibodies which are added to enhance the reaction. Once it is incubated for a particular period of time, it is run through the flow cytometer. Once it is run through the flow cytometer, you can see a shift if there is a reaction. So if you see a right shift, you know that this is positive. If you see a left shift, you know it is negative. Why? Because more the reaction, the more the binding, and the spectrophotometer will give you a reading and the graph will change. This was considered to be very, very sensitive, but it is a difficult test to establish. Hence, many of the laboratories all over the world still do not have this, but major academic centers have managed to establish it. So now, how does CDC compare with the flow cytometer? So if you see a CDC cross-match, by the time you have a neat serum, it is positive. If it has a lot of antibodies, but by the time you dilute the serum to 1 in 8 or 1 in 4, the normal CDC starts turning negative. So if you have diluted the antibodies and the antibody titers are coming down, the CDC turns negative. Then you add AHG, anti-human globulin, because you give more arms for reactions. Then till 1 in 16, it will still remain positive. So you will catch hold of more CDC cross matches which are positive, and you will identify more people who have antibodies. Flow cross match will remain positive till 1 in 64 dilution. So here, how does the MFI match? So 10 to 15,000 MFI of an antibody will give a flow cross match positive. So still it is on the higher side. What about antibodies which are 3,000 and 5,000 MFI? They will still not be caught up in this. So we need technologies which can merge with each other and give us information. And when the flow cytometry came in, there were myriads of papers which came in, and they said that if you have a flow cross-match positive, which is a very sensitive platform for cross-match, your early graft loss is very high. So if you have a flow cross-match positive, please do not go ahead. So if you see, most of the studies, including the one till 2001, the early graft loss is 33% if your flow cross-match is positive versus 11% if it is negative. So CDC slowly is getting replaced with flow, but there are centers who are still using both the technologies to marry each other so that you can get more robust information. So in our case, retrospectively a flow cross match was done. The CDC was negative, remember, but the flow turned positive. That means that the patient actually had antibodies, but they were of a lower titer which the CDC did not pick up. Now, can you still think that the flow might give you non-HLA antibodies? It is giving some false positive, false negative. So you need something more robust, because in today's modern science, you need everything to be black and white as far as possible. So then solid phase assays came by. Here, the advantage is that only HLA antibodies will be identified, unlike cross matches where non-HLA antibodies will also be identified. Only IgG antibody, which is relevant to transplant, will be identified, not IgM. IgM means autoantibody, not relevant to transplantation. CDC crossmatch might pick up IgM, and we can negate it with DTT, but it can confuse. A solid phase assay can reliably determine antibody specificity. Now, when you're doing a crossmatch, you don't know which antibody has reacted and why it has turned positive, whether there is actually an HLA antibody or it is just false positive, which also happens. So, it can reliably tell you which antibody is present and whether it is similar to the antigen on the donor or whether it is same as the antigen on the donor. So, you can easily match the profiles. It will help you avoid immunologically high risk transplant. So today, if I know that my donor has A24 antigen and the recipient has an A24 antibody, I might want to avoid that versus I might want to negate the antibody before I go for transplant. I might be able to find an appropriate donor if he has more options of donors rather than do an immunologically high risk transplant. In these solid phase assays, a body-like system is not required, a complement activation is not required, so not much of a problem. And important, it can detect very low level antibodies also, which will be missed by the cross matches. So solid phase assays have a lot of terminologies and many people get confused interchangeably between these. So you have something known as panel reactive antibody, which is just a screen. So you are screening the blood saying yes or no, whether this patient has antibodies or no. And if yes, how much? So PRA percentage means how much percentage of population you are reacting to. So a measure of antibodies in the patient against a panel of antigens from multiple donor cells from the population, as opposed to only one donor when you do a cross-match. It gauges the immune reactivity of a patient to the population. So if you have a common HLA antigen against whom the patient has antibodies, he or she will react to multiple donors in that population. And that is how you calculate the calculated PRA. So you come to know how much is the chance of the patient getting an organ. Because if you have a common antigen against whom you have an antibody, then you will not find a donor very soon because the same antigen will keep repeating in many patients, many donors. These have to be monitored 3 to 6 monthly because there is an antibody wax and wane in our circulation based on the release of memory cells or not. Every stimulus gives an exposure to the foreign antigen and it might be stored in the memory cells, which might come out with, say, viral infection or a re-stimulus. So we need to keep doing this 3 to 6 monthly. Once you have done a panel reactive antibody and it turns positive, then you need to identify that which are these antibodies which is giving the PRA positive, whether it is 1%, whether it is 10%, 2% or 100%. You need to know which antibodies, you need to identify them. So these are the solid phase assays available to us. One is the screening beads where it is like a PRA. So I am going to say yes or no. So I am going to run the serum of a patient with a bead who has multiple antigens attached to it. And the antibodies, if present in the patient's serum, will bind to this bead, the antigens on the bead, and it will show a reaction. It might not show which antigen it reacted to, but it will show which beads it reacted to. And we might say, okay, there is a 0 antibody or there is a 10% or a 5% or a yes and a no. Class 1 yes, class 2 no, etc. Now I want to identify which are these antibodies. So that is where single antigen bead assay comes in. So you have a bead where a single antigen is attached. So there is a bead which has only A1 on it. So now when the serum of the same patient is run along with single antigens. So every bead will have a single antigen attached to it. And all the antigens are then represented on these beads, and each bead will only one specific antigen with a density on it. So once I have a reading of a single antigen bead assay, I will be able to tell you which are the antibodies present in the patient's serum. And this will then tell me whether these antibodies present in the patient's serum are going to be specific to a particular donor or not. This is what calls for a virtual cross-match. So I am doing a cross-match with a panel of antibodies which have turned positive on the patient's serum, and a donor's HLA is being matched with it. Now there is some complexity in it because an HLA antigen will have a lot of cross-reactive epitope groups. So you do not match numbers to numbers, you match families and numbers. So which is a little complex, but that's okay. The laboratory, we run the laboratory, we do it for you. This is how a typical run looks like. So if you see, the red bars have an MFI of 14,000, 12,000, 11,000. So these are the antigens which have spiked positive, and these are very strong. Then you see these are the orange ones which are moderate. So these are around 2,000 to 4,000 MFIs. And then there are questionable which are around 1,500. So usually 1,000 to 1,200, 1,500 we call them indeterminate. Then we have to mix and match what is the CDC cross match, what is the HLA antigens on the donor, how reactive it is, and then we give out the report. But this is essentially how a run looks like. And these are the MFIs. So it is very easy for me to identify and tell you that these are the analysis and the antibody specificity. So now if you see from a CDC to a Luminex. So in the CDC cross match, you need very high level of donor specific antibodies to give it positive. And as the technology advances, even the low level of antibodies will be picked up by a single antigen bead assay till the DSA is negative and it becomes negative. So this is the technology for you which has come up from the year 2000 to 21. In the last 2 decades, the technology has just revolutionized our way we think of a transplant. So now case 1, the final analysis. So we saw that the CDC cross-match was negative. Retrospectively, 4 cross-match was positive with the current and the remote sera. That means the patient always had low titer antibodies. Retrospectively, PRA was done because the technology was not available at that time. 5% to class 1 HLA antibody was found. Then we looked at the HLA mismatched antigens and the A23, B37, 53, DR beta 103, these were mismatched. So now I have to look at antibodies in the patient against these mismatched antigens. And A23 was identified as positive in the single antigen bead assay. So conclusion of the case, A23 was a donor-specific antibody which caused a humoral rejection and graft loss. This was a preformed antibody, although in lower titers, and as soon as the graft went in, it attacked. So that is why only a CDC would not have been enough. Enhanced CDC, a flow cross-match and an antibody analysis would have given us the answer, and we would not have gone for transplantation if we would have had the technology at that time. So the virtual cross-match technique, like I told you, it refers to the comparison of anti-HLA antibodies of the recipient as defined by a single antigen bead assay with the HLA of the donor. If there is a donor-specific antibody present, this would represent a positive virtual cross-match. So we are not doing any test in the lab. We are just looking at the antibody profile and the donor HLA antigens, and we are doing the cross-match in our brain. Antibodies are defined against HLA class 1 and class 2, and these are then identified as donor-specific or non-donor-specific. So now what is the clinical paradigm we have today? So the immune threshold is moving. If you have a CDC negative or an AHG negative and a solid phase negative or a flow negative, your virtual cross-match negative, you go ahead with the transplant. If you have a CDC positive, AHG cross-match positive, flow positive, virtual positive, then the transplant most probably is contraindicated, but you can proceed with caution with desensitization or with acceptable mismatch. What is acceptable mismatch? That you try to avoid a donor against whom the patient has antibodies. So suppose one donor has an antibody against his antigen in the patient, you try to find another donor for that patient and you can go ahead with the transplant. So the point is you avoid the antibodies, you accept the antibodies, but you bypass the antibodies, or you remove those antibodies. That is the two ways you can go ahead, but you will have to evaluate the patient thoroughly. So the take-home points: mismatched HLA antigens will act as a potential stimulus for rejections. Typing will define the magnitude of the potential stimulus. Accurate typing is imperative for patients who already have HLA antibodies prior to transplantations. Molecular typing is recommended. Antibody screening with PRA, pre-transplant, and identification with SAB, if screening is positive, will help you tailor the immunosuppression, will help you choose a suitable donor against whom the patient does not have antibodies. Very important. This is the concept of acceptable mismatch. And a CDC crossmatch defines the risk of hyperacute rejection. A flow crossmatch defines the risk of early immune complications. A crossmatch must always be interpreted with antibody screening data for relevance to that transplantation. And an HLA laboratory is just not a laboratory. It is risk assessment for your patient. Be in regular touch with the laboratory person, because you can treat your patient better. And I'm lucky that being a nephrologist, I get to see both the sides, that in the laboratory also, in the bedside also. And I know what a clinician goes through when things are not right. And with that, I think I will end. And we can do some question answers or discussion.
Wow, that's very good presentation. Thank you very much.
Thank you.
Yeah, very exciting and educating presentation. In Africa, transplantation still is new. I know. Especially in East Africa, with the exception of Kenya, Tanzania and Uganda, Rwanda, the transplant is quite new. So there are a lot of, I'm quite sure some of us here, there are a lot of things that we think they are kind of new to us. Yeah, so it's a question time and comment, but as usual, let me start with the first question. So now, to me, in Tanzania, we started, I think, transplantation in 2017 or 18. I don't remember very well, but we do both CDC and flow cytometry. But as per the presentation, it seems, CDC actually has less sensitivity compared to flow cytometry. Yes. So does it mean that if you don't have CDC, you can only, and you only have flow cytometry, you can only do that and you won't be worried?
It would be worse. You can do only flow cross match, but you have to have antibody profile along with it, so that you can add it together. Sometimes the flow cross match is a very sensitive platform.
Okay. So PRA must be done in that particular case.
Yes. And if the PRA is positive, you need to identify whether it is against the donor or no. If it is not against the donor and the flow cross match is positive, you can neglect it. Okay, so it is important to tie the technology together.
Thank you very much. Thank you very much.
And CDC is very simple to have. So I would say, even if you don't have flow cross match, the CDC cross match is more important along with an antibody assay if you have that.
Thank you. Thank you very much. Yeah. Any other question or comment from the audience?
I think Dr. Bhima has a question. He has raised his hand.
Yeah. Thank you very much, Ajan. Thank you, Dr. Tapiawala, for that wonderful presentation. It was really, really, really very good. So, as Ejana said, we are all pretty much dilettantes when it comes to transplantation, but in South Africa, we've been doing it for a while. But in my center in Durban, we only started a few years ago. So we are still very much on the learning curve. So what I wanted to ask about is, can you just explain to the audience about mean flow intensity, because that is something that always comes up, and at what level would you take a cutoff? Because our surgeons and I have a difference between what we consider to be suitable and what's not. But I'd like you to explain the concept to the audience first.
So when I showed the graph, the single antigen bead assay graph, if I may share again, I can show it to you. Yeah. So if you see this, this is the MFI. So that means
Yes.
mean fluorescent intensity means the strength of that antibody. So if you have a very strong antibody, it is going to very strongly bind to the recipient antibodies or the donor antigens and cause a strong rejection response. If you have moderate to questionable antibodies, we might get away without getting a rejection with just mild plasma exchange and immunosuppression. Now, what MFI is significant? So, in my lab, so every lab has to identify its own cutoffs with what you will call it positive, what you will call strong, and what you will call weak. So, in my laboratory, more than 5,000 is called as strong, 3,000 to 5,000 is known as moderate, 1,200 to 3,000 is called weak, and below 1,200, 2,000 is questionable. So I would say if you have an antibody MFI, which is more than 5,000, but you have to stick to only your lab. Because see, what happens, you cannot compare the MFIs of one lab to the other. It's not like creatinine. Like, even if you see creatinines, if the ranges change, your creatinine level changes. Similarly, the MFIs keep changing. So if you run the same serum in two different labs, in one you may get 6,000, one you may get 10,000. So I would say that you have to discuss with your own laboratory person that how strong do you feel these are. If that person says, no, this is 6,000 in my laboratory, above 3,000 is very strong, then you take it as strong. So what I would say is largely more than 5,000 is usually regarded to be strong. And I would say that anytime you have a donor specific antibody going ahead for a transplantation, at 8-10 days, you might encounter a rejection. I don't know how often you have seen this, that if you have a donor specific antibody, you have gone ahead, within a month, you will see the effect.
Absolutely. Okay. So, we are looking at, I usually take a figure of anything above 2,000 as being the cutoff from our lab. But the surgeons are happy to go ahead with 3,000 to 4,000, which then leads us to a problem.
Yeah, but then you will have to precondition the body.
Yes.
You will have to precondition nicely. And the problem is that these antibodies are going to cause you a niggling chronic rejection later on, because they are not going to keep quiet. Yeah. These are going to keep circulating later on also. You will remove these during the transplant. After that, what? So I try to avoid people having DSAs. I will not tell them no transplant, but I will give them donors. I have a registry for sensitized patients. What do? I swap donors. I do a paired exchange wherein I give them donors against whom they don't have antibodies. So they will get a transplant. They might wait a little bit more, but they will get a kidney with a low immunological risk, because I don't want to keep doing plasma exchange and over immunosuppressing them all their life.
Absolutely. Thank you so much. That was very helpful.
All right. Yes.
Something about excellent presentation, Shruti. Wonderful. It is very clear and precise. Very down to earth presentation. So understandable. Can you tell us something about your registry and the paired exchange program that occurs because of the registry?
Yes.
It will be because Mumbai leads the country in the paired exchange programs, and they've also done domino transplant. So can you please just explain that to the audience?
So there are two types of registries. One is for a blood group mismatched transplant. So now, as the entire world knows, blood group mismatched transplants are done easier than said, and the outcome is as equal as ABO in compatible transplants. So the registries initially, like Astra registry, was set up in Mumbai by the Apex group, wherein they were matching blood groups and giving donors. That was for the paired exchange. Now I realized that blood group incompatible transplant is much safer than doing an HLA incompatible transplants. And since I was trained in Toronto and we used to export these patients to an LDP registry, I thought of setting it up in India. So we have a registry known as SORTER, SORTER. So SORTER is Sensitized Organ Recipient Exchange Transplant Registry. What this does is that we list the patients who are highly sensitized, so who have donor specific antibodies against their donor, who is fit enough to give him or her. Then I will gather all, I have around 15 patients just now who are active on the list. And what I do is every time a donor is added, I will match them with the previously present recipients if they have an acceptable mismatch. So they will not get an HLA match, but they will get a donor against whom their blood or their serum does not have antibodies. And we have done 6 such pairs till now. Not required plasma exchange, they have not required thymoglobulin, they have gone ahead with basiliximab. So because I chose to give them a kidney against whom they don't have antibodies, so it's like a big boulder in front of me and I just bypassed it. So that is a sorter registry. So if we have sensitized recipients, we can use this registry. There is an IT team looking into making it as a electronic version, but it is at the last phases where it can start running on its own and doing runs. Because doing a blood group mismatched automatic run is easy, but doing an HLA acceptable, I have to look at cross-reactive epitope groups. I have to look at families of the HLA antigens. So, you know, it's taking a little time in the end for them to put it together. But till then, I'm doing it manually.
Thank you. Thank you so much. Is there any
Dr.
other question? Yes. Yes,
This is Dr. Wala.
Dr. Wala, thank you. Thank you for coming.
Thank you very much.
Yes.
Thank you for the presenter for the excellent presentation. As a follow-up for the MFI question, it always keeps on coming up again and again. We as medics, we like a uniform way of doing things, and if one lab is giving a cutoff of 3,000, other one at 5,000, as you have mentioned, is it possible for the labs to have a uniform way of reporting the MFI?
That's what I said, that every laboratory has to identify its own cutoff. So say if you send the sample to me in India, I will be able to tell you that this is a strong antibody, this is the cutoff we are using, this is strong, this is moderate, this is weak. So you should go by that and not by the MFI. So if your lab says this is strong, you take it as strong, because you know the reactions which come in the system can be recorded. So what I have done in the lab is that we have run negative samples. We have seen what is the negative values, then taken a 3 standard deviation up and 3 standard deviation down. So if I have a 3 standard deviation up, I say it is positive. Then after the 3 standard deviation positive, that becomes weak. Then 2-3 times that becomes the strongest antibody. So every lab does that for the cutoff. The factory made brochure of the Luminex machine or the One Lambda labs or the Immucore labs will give you a cutoff of 1200, and they will then say you grade the antibodies based on it.
And so don't you think it brings a lot of subjectivity as to
it does, because every country has a problem unifying it, and it is concluded everywhere that you cannot have one laboratory give the same value as second, because proficiency testing fails in this.
Okay, thank you very much.
All right. Dr. Wala is a consultant nephrologist from Aga Khan in Nairobi and is part of us as well.
Okay.
Thank you, Dr. Wala.
So there's a question in the chat box. What cross-match would you do for a deceased donor? Is it both virtual cross-match and CDC before transplantation, or virtual is enough? So there are robust programs in Russia and in Europe and Germany where they're using only virtual cross-match. But for that, you need a robust system where you have 3 monthly samples which are collected and a cumulative antibody list with you. So for now, for 99% of the countries, a virtual cross-match along with a CDC or a flow is enough. Like Toronto Medical Labs where I work, they have done away with the CDC cross-matches. They do flow cross-match and they do a virtual before a deceased donor transplantation. So I don't think we are at a time where we should go only with virtual unless our program is doing more than 500 transplants or 1,000 transplants a year and our institute is very robust in transplanting.
Thank you very much for the nice presentation. I'm Dr. Mgume.
Yeah, hi.
Yeah, thank you. It is really educative and we really followed it up in a good way. So for Tanzania, we started the kidney transplantation at Mumbai National Hospital November 2017.
Okay.
I wanted you to clarify more on just the HLA match. For example, if there is a mismatch of, if there is haplo match,
Yeah.
Sometimes we go up to 2/6 match. So
yeah,
any impact, because for 2/6 we do the donors we take and the recipient, and for tests that we do currently, we do CDC and flow cytometry.
okay, okay.
So what is your comment on the
So
outcome? For example, we decide to do
so with the flow cross match, so with the flow cross match that you are doing, it is excellent because it gives you that margin of picking up low titer antibodies. But if you have a PRA screen, nothing like it, because I have seen flow cross match is negative with 5,000, 6,000 antibody levels, and you will still have antibody mediated rejections higher than when you do a solid phase assay. So as soon as possible, you should try to do solid phase assays on the patient. But till then, what you're doing is good. Continue that. But as soon as you can get the technology for your patients, get it.
Oh, thank you very much.
I think, Jonathan, I think, so Dr. Jonathan runs a program in Mumbai National along with Dr. Shaw.
Okay.
So I think, Dr. Jonathan, I will get you in touch with Dr. Shruti. And these things, I think if you discuss a little bit, it will be very helpful. And I'm sure Dr. Shruti will be very, helpful and eager to help out as well. Yeah.
Sure.
So I think I will connect you, Dr. Jonathan. So it will definitely help you a great deal.
All right. Thank you. I think we have come to an end of today's presentation. I should thank the presenter, Dr. Shruti. That was excellent presentation and very educative. And also I should thank Dr. Lloyd, who has worked really hard to get Dr. Shruti on board for today's presentation. And also I should thank all of you who attended this presentation. So thank you very much. So we'll meet next week.
We hope to have you on board again, Shruti, for many more sessions.
My pleasure. And it was a pleasure talking to all of you and interacting. And it's always interesting to know different perspectives from everyone. And I'm always there to practically help you all out, case-to-case basis. I'm sure Dr. Lloyd will share my number and email address with you all if required.
All right. Thank you. Thank you. We have come to an end of our presentation today. Thank you very much.
Thank you very much.
Dr. Shruti.
Thanks.
Yeah,
Bye-bye.
we really appreciate it. All right. Bye-bye.
Thank you very much.